So to give you an example, just to really highlight this, a number of years ago I lived in Belize for about 2 months, and I was surprised to find that there were no individuals with type 1 diabetes around. And the reason was because the availability of insulin was very erratic. And so there were periods of time when insulin just was not available. So unfortunately, people with type 1 diabetes passed away during these periods of lack of availability. So type 1 diabetes requires insulin to shut off acid production in the liver, which can be fatal quite rapidly. So we still don't understand what are the primary inciting events for type 1 diabetes. Active research includes understanding the immunology. What is it? What's the specific antigen that's activating T cells that are contributing to the immune response? And asking questions, can we prevent the immune response, or can we allow insulin-producing cells to survive despite this immune response? There's also active research programs going on with islet cell transplantation, immunomodulation, other factors. But for right now, people who have type 1 absolutely require insulin. And I think that it's not that long ago that insulin actually was discovered. And it's hard to remember, but prior to insulin, individuals with type 1 diabetes were kept alive by a starvation diet, no carbohydrates, high fat intake. And as you can see in this child, they were very emaciated. And then insulin was really a miracle cure that allowed people to survive during childhood. Of course, there are still complications of type 1 diabetes and with type 2, but including eye disease, heart disease, kidney disease, and nerve damage. So it's still a very, very serious disease that has to be cared for very well, but absolutely requires insulin. Okay. Any questions about type 1? Okay.
What did they find with the islet transplant? Was that successful at all, or different results, or?
So islet cell transplants are very effective. If you restore insulin production, you cure the defect that's present. The challenge is keeping the transplanted cells alive. And so, the immune response is the big issue. There's two issues. One is you're transplanting cells from someone else. So obviously there's an issue with the donor versus the host. And then second of all, you're transplanting islets typically in a place, for example, in the liver, that's not physiological. So there's some issue about dysregulation of how insulin is actually secreted. But the main issue still is one of persistent immune response that's contributing to failure in the long run. Now, there are some people where that's not been a problem and have been quite successful. Also, you can transplant entire pancreases, and that's usually done in the setting of a kidney transplant at the same time. And in some cases, they're very effective. People have long-term survival of the donated pancreas, and they're off insulin. So it can be extremely successful, but the major issue is still the immune response. Okay, so type 2 diabetes is more common, 90% of all diabetes. As I mentioned, previously called adult onset, but not the case any longer. And the major issue here is not that the body doesn't make insulin, but that it's resistant to its actions, or insulin resistance. So there's plenty of insulin initially, but the body doesn't respond well to it. So let's say that I inject a certain amount of insulin, my blood sugar might drop by 50 points. I'm just making something up. If someone's insulin resistant, they might have less of a response. The blood sugar might drop only 20 points. So resistance to the normal action of insulin to lower blood sugar. So initially, the pancreas compensates. Insulin levels go up to compensate for this block. But over time, there's loss of insulin secretion by the pancreas, and ultimately the glucose goes up. Okay. So people with type 2 diabetes, even when their blood sugar is elevated, still make some insulin. Not like type 1 where they make zero insulin. So here there's still some insulin around. So given that there's some insulin secretion, individuals with type 2 diabetes are not prone to developing acid production by the liver, ketoacidosis. So even if they're on insulin, if you withdraw the insulin for a short period of time, blood sugars will go up, but generally people won't die from insulin deficiency. Okay. And finally, it's often treated with medications other than insulin initially. First diet, lifestyle changes, medications, and perhaps ultimately insulin. Okay? Okay. So from here on out, I'm going to be talking about type 2 diabetes only. If there's any other questions about type 1, we can get back to that. So I think it's really important to consider people at risk for diabetes. Ultimately, we'd like to prevent diabetes rather than wait for it to happen. And type 2 diabetes is quite preventable. A number of strategies are effective. So we'd like to identify people at high risk, educate them, and try to initiate some lifestyle changes that will reduce their risk. So, some of the risk factors. Family history of diabetes. Well, this is actually a very strong risk factor, and it's actually a stronger risk factor than for type 1 diabetes. And some of it's genetic, and some of it's shared environment. If a family is eating healthy and has an active lifestyle, their risk will be lower. So some of that's genetic, perhaps, but some of it's also shared environment. It's important to think about. Ethnicity is also important. What ethnic groups are at higher risk? Does anybody know? African Americans, Hispanics, Native Americans, Asian Americans, both South Asian Indian and also, for example, Japanese Americans. Birth weight. I hope you all know your birth weight. If not, go ask your mom or look at your birth certificate or something. Interestingly enough, the intrauterine environment is a really important time for initiating risk for diabetes. And we can't really study the intrauterine environment in humans, but the birth weight is kind of a readout of how your intrauterine life went. And so if you're low birth weight, you have an increased risk of diabetes. And conversely, if you had a high birth weight, you have an increased risk of diabetes. So the best thing is to be right in the middle.
What would be that range, the right in the middle?
So generally low is less than 3.5 pounds, and high would be greater than 8.5 pounds.
What's high? 3 is high.
A lot of the risk with high birth weight is attributable to the mom having gestational diabetes. If the mother had diabetes during the pregnancy, the high glucose levels actually cross the placenta and stimulate the fetus to make insulin and other hormones that actually increase growth and increase fat deposition. And that sort of sets off a cascade that's persistent during adult life. So from a public health perspective, this is really important because, as moms become more obese, they're obese before the pregnancy, there's a higher risk of gestational diabetes. Then the babies are at higher risk because the mom had gestational diabetes. And so it's a vicious cycle, and we've really got to work to decrease this. Another major risk factor is obesity, particularly in the abdominal location. So for anyone who is concerned about fat rear ends, that's not a particularly big risk. The big risk is abdominal fat, beer bellies, whatever you want to call it, is really a dangerous risk factor because the fat within the abdominal location has a much more deleterious effect on insulin action than fat anywhere else. So abdominal fat's not okay. If you had a history of diabetes during previous pregnancy, you're at higher risk. We mentioned that before. Increasing age, 45 is really very arbitrary. It's really just increasing age, huh?
Can't avoid that.
Can't avoid aging now. High blood pressure, high lipids, and importantly, inactivity, all risk factors for diabetes. Okay, so I can't resist showing this because I love this picture. I think it's just a great example of every possible reason. You know, only the dog here is getting enough exercise. Mom's overweight, father looks just like him. He's got the big belly, he's drinking a soda, maybe some chips down here, so perfect. And then, I don't know how many of you saw WALL-E, but I thought this was such a great, satirical commentary about what's going to happen to us if we don't change our lifestyle and get together, because here we have central obesity, clothes don't fit anymore, and they were so inactive that it was really striking. So I'd bet he has diabetes. So diabetes is really, type 2 diabetes is really a disorder of multiple tissues. And so, for example, in muscle, there's insulin resistance. And in muscle, insulin normally causes glucose to go from the bloodstream into the muscle. So if you're resistant to insulin, not as much glucose goes into muscle. Same thing happens in adipose tissue. But in addition, adipose tissue is not just a storage depot for fat. It's really an active organ that contributes to helping your body assess its nutrient stores. So for example, if your adipose stores are very low, your body won't make enough leptin, and you'll be stimulated to eat more. So there are really a number of different hormones that come out of fat tissue that are important in regulating metabolism. Another important organ is the liver. The liver is key for making glucose. When you fast overnight, hopefully you're not eating, right? Maybe some people are, but hopefully not.
Sleep eating.
Sleep eating, right. Well, actually that happens. It does. But you're not eating. So if your liver didn't make glucose, your blood sugar would drop overnight. So this is a very, very important function of the liver. But in diabetes, it's gone wild and it's making too much glucose. The pancreas is also important because insulin production is inappropriately low. It's not zero, but it's still not enough. And there are changes in some other hormones like glucagon and incretin hormones that actually come from the intestine. And then finally, there's more and more information that's still emerging about an important role for the brain. Brain is really what controls our food intake. Do we feel full? Do we want to eat? What stimuli are making us hungry? And these may be altered in people with diabetes and contribute to differences in eating patterns. Also the gut. The gut is very important because the gut actually makes hormones that help regulate insulin secretion and appetite too. And a really interesting finding recently is the role of gut flora. So we all have bacteria in our gut, and each of the bacterial profiles in each of us is different and can affect the metabolism of nutrients in our intestine. So a lot of things, and so we really don't know which of these is primary. So I think of diabetes as a big puzzle. And from a scientific perspective and a clinical perspective, there's a lot of unanswered questions. So, this is not a simple question to be addressed. One of the simpler questions is which comes first, insulin resistance or decreased insulin secretion? I think the answer there is probably pretty straightforward. Most studies show that people have insulin resistance for years, and only when the pancreas can't compensate does the blood sugar rise. So by the time people actually get diabetes, this process has been going on for a long time. And there's a big problem with that. When you have insulin resistance, even without diabetes, your risk of heart disease goes up. Insulin resistance is associated with clogging of the arteries or atherosclerosis. So you don't have diabetes yet, but that process is still going on. So that's another reason why we're very concerned about obesity, because obesity causes insulin resistance, and insulin resistance causes asymptomatic atherosclerosis, even before diabetes develops. So I think, let's see. I showed you a lot of tissues that contribute to diabetes, but we really still don't know which of those tissues is the first defect. You know, is it really the brain as the primary defect? Is it really the muscle or the adipose tissue? We just don't know. What we'd like to know are what are the molecular problems causing insulin resistance in individuals at high risk prior to the development of diabetes. And then, of course, if we knew that information, could we use this to improve treatment and prevention? So, I mean, that's the ultimate goal. So this is a slide which kind of summarizes some of the things we've talked about, and I just want to go over it. If you think of this timeline as the 0 point is when diabetes is diagnosed, what's really clear is that insulin resistance has occurred for many years prior to the onset of diabetes. And this information has been gleaned from a number of very large epidemiologic studies where they've just serially followed people in populations. And by backtracking, can show that these changes existed many years before diabetes. And so insulin resistance increases, particularly with obesity, and is present when diabetes is diagnosed. Concurrently with that, insulin secretion goes up to try to compensate. But at some point, the insulin compensation is no longer effective, and insulin levels fall. And that's when glucose levels start to rise. Again, I want to make the point that the macrovascular changes, which include all the blood vessel atherosclerosis, starts at the time when the insulin resistance is developing before the diagnosis of diabetes. The microvascular changes, which are eye, kidney, and nerve damage, that only starts once the glucose itself is elevated. And it's thought that the glucose elevations are what's responsible for those complications. Okay, so the problem with diabetes is that it's really been there for years before it's diagnosed. And it's this insulin resistance is intimately linked with obesity. So obesity is really one of the most common underlying substrates and is a disease.
Are there any signs of insulin resistance before you actually get diagnosed?
Well, there's some clues. So obesity itself is a pretty good clinical indicator. Waist circumference is a good indicator because it reflects the fact that it's that abdominal fat that's particularly dangerous. If you look at the lipids, there are clues from the lipids. If your doctor draws a lipid profile, elevations in the triglycerides and low HDL or good cholesterol. These are all signs have been recently released which doctors can use to quantify insulin resistance. The difficulty is that most of the time we do this in the quantification of insulin resistance in a research setting. So it's just recently that some of these tests have been available for clinical practice. But the reality is that body mass index, waist circumference are really pretty good markers of insulin resistance. So everybody has access to those.
I'm not really sure on the macrovascular changes versus microvascular changes. Is the macrovascular changes the atherosclerosis that begins, and the micro is the downstream effect of those on the organs that are getting lower?
So macrovascular changes refers generally to larger arteries in the body which become clogged with lipid deposits, so atherosclerosis. So those would be the kind of changes that lead to heart attack, strokes, peripheral vascular disease, where people ultimately would require amputations in some cases. Those are all larger blood vessels. Microvascular changes are those changes that occur typically in smaller arteries that appear to be linked to high glucose. And the most common places where you see evidence of that are in the eyes, which can cause blindness, in the kidneys, which can cause kidney failure, and in the nerves, which can cause neuropathy or loss of sensation in the legs. So macro is atherosclerosis, microvascular are the end organ, what we call end organ complications of diabetes, the eye, kidney, nerve damage.
Do they know why glucose causes these? What's the physiologies of why these acts on the blood vessels?
Yeah, well, high concentrations of glucose, that if you have a cell and you add high concentrations of glucose or an animal with tissue, several things happen. There's a lot of oxidative stress that changes signaling within the cell. The high glucose concentration itself can be converted to polyols, and high glucose activates different kinases within the cell, for example, protein kinase C. And this can trigger a cascade of changes within the cell that affect the response of the endothelium, for example, to normal hormonal stimuli. So it's, in fact, a number of different medications have been tried to target glucose, the effects of glucose on cells, with limited amounts of success. Among the most promising are these inhibitors of protein kinase C that actually appear to normalize cellular function, and there are ongoing clinical trials to look at reversal of this pathway in particular. So, okay. So, what are the earliest abnormalities in individuals at high risk? And if you take, this is data now from 20 years ago, where they actually studied how people responded to insulin. So these are healthy controls, and they had a normal level of insulin sensitivity. Just let's say this is normal, okay? We won't go into the specifics of how the test was done. But in individuals who were relatives of patients with diabetes, there was already a substantial impairment in insulin action. And of course, individuals with established diabetes had an even greater decrease in insulin action. So what this kind of information says is that insulin resistance is present in individuals with a family history of diabetes long before glucose has become at all abnormal. And so it's an early defect. So what we'd like to ask is, what are the patterns of gene expression in key target tissues in humans with established diabetes or those at risk for diabetes? Can we identify defects that are occurring before the diagnosis of diabetes? I'll tell you a little bit about some of our work addressing what's called the mitochondrial hypothesis of diabetes, and a little bit, if we have time, about other pathways associated with insulin resistance. So, the central question that we'd like to try to answer, to give it a framework, is, okay, if insulin resistance is an early defect, and impairments in insulin secretion caused by beta cell dysfunction are both required for diabetes, can we walk backward to identify changes in gene and protein expression and function that are responsible for both of these defects? Not an easy answer, easy question to address, mostly because we're looking at individuals who don't have a disease, who are just at risk for a disease. So what are some of the issues that you might anticipate from this kind of approach?
They might never develop.
They might never develop the disease. You can say, okay, you have a family history of diabetes, but it doesn't mean that you're the one that's going to get it. You're at higher risk, but it's not 100%. Right, exactly. What else? So if you don't have a disease, the defect is likely to be fairly subtle. And we're looking for changes in people who are otherwise healthy. People who are at risk for diabetes, walking around, exercising, maybe not as much, but their bodily functions are normal at any gross observation level. So it's very subtle. It's not like when you're looking for cancer defects where you have a normal tissue and a very abnormal tumor. So this is a very subtle difference. Yes?
I'm going to ask the question backwards. If the person who's at high risk, and let's say they are going to get diabetes at some point in their lifetime, and you're looking for that gene and protein expression change at some point in their life and where it is, but they do all the things right, reduce their weight, get lots of exercise, that implies that that has an effect on the gene expression. So would you miss it in a person if you could know this person was going to get diabetes, but they could stave off that diabetes by
Absolutely.
these altered changes?
Absolutely.
So the changes back, the changes in lifestyle change back the protein gene expression.
Right. But there's still an underlying substrate, genetic substrate, that probably makes that individual respond differently to an environmental stimulus. They're doing all the right things, and so it's going to be more difficult to detect that signature in those individuals. So one of the things that we do is actually metabolically characterize individuals. So we know that this person has an insulin sensitivity value of 10, and this person has an insulin sensitivity value of 5. I'm just making up numbers.
Right.
So now we can say, okay, what changes correlate with this metabolic abnormality? Because that's a way to try to get around that, because that person who exercises all the time to try to prevent diabetes will actually be more insulin sensitive than another high-risk person who never exercises. Does that make sense?
Absolutely. So it seems to me that we have a lot of candidates out there for research.
Yeah, absolutely.
I mean, if the risk factors are so well known, I mean, gestational diabetes, high birth rate, family history, all of that, then it seems to me that we have a plethora of people to study
Well,
from.
if you can encourage them to sign up for research studies, it's a little more difficult to get them to enroll in research studies. So each of these risk factors could alter gene and protein expression and function via different mechanisms, right? Doesn't necessarily mean that they're all working through the same mechanism. Ultimately, they may all cause insulin resistance, but at a cellular level, the way they get there can be completely different. So, for example, if you have either some sort of adverse intrauterine environment, either poor nutrition or poor function of the placenta, it can actually change your development, okay? Or it could affect gene expression through epigenetic changes, which I don't have time to get into. So that's a possibility. Family history could be genetics, or it could be shared environment, as we mentioned earlier. Well, we can't change aging, but aging has effects clearly on physiology as well. And then down on the bottom are what people traditionally think of as environmental factors. Energy excess, dietary excess, obesity, and inactivity. And there have been a number of studies over the past few years implicating a number of different pathways in contributing to these effects, including abnormal function of mitochondria, accumulation of fat within tissues. So, accumulation of fat in muscle or liver, it doesn't belong there. Fat belongs in fat tissue. And if it starts accumulating in your muscle or in your liver, that's very damaging and causes insulin resistance. Oxidative stress is another big candidate factor for mediating insulin resistance. Abnormalities or stress within the endoplasmic reticulum, and also inflammation can all contribute to insulin resistance. So let me just tell you the strategy that we take, and there's no perfect strategy whatsoever. This is what we've tended to do in our studies. So if you think of this as a continuum in individuals who are insulin sensitive, insulin resistant, and then some of whom go on to develop established diabetes, we'd like to study how the disease progresses over time. As you might expect, longitudinal studies are very difficult. So what we have done instead was cross-sectional studies. looking at insulin-sensitive individuals, insulin-resistant subjects, and patients with established diabetes. In our studies, we've actually looked at muscle and adipose tissue biopsies from metabolically characterized volunteers. So we assess their insulin sensitivity and then do biopsies of their tissue. And then we've used the tissue to assess gene expression, protein expression, levels of metabolites within muscle, and view those types of data as a way to integrate genetics and environmental effects on the cell. And then, of course, when you identify a candidate gene or pathway, you have to test whether this might influence metabolism in either animal or cell culture models. And that's kind of one of the strategies that we and others take. So what have we learned from these approaches? A lot, still ongoing, and I just want to briefly touch on one result, which is that many studies using a variety of techniques have shown that type 2 diabetes is associated with alterations in the function of mitochondria, in muscle, in adipose, and even in liver tissue. And our studies, as well as a number of other studies. So I know you're all familiar with mitochondria, but mitochondria are just really important for ensuring normal metabolic homeostasis. As you know, nutrient fuels in the presence of oxygen are used for the generation of ATP, and in the process, water and heat are also produced. Mitochondria are controlled essentially in two different ways. They have their own mitochondrial DNA, which accounts actually for a minority of proteins within the mitochondria. Most are actually encoded by nuclear DNA, and proteins are made in the cytosol and then get imported into the mitochondrion. So there's this very important crosstalk between the mitochondrion and the nucleus that seems to be important for maintaining healthy metabolism. And in one of our earliest studies looking at gene expression, we looked at muscle tissue from individuals with diabetes, or with a diabetes family history, or with no family history. So this is a heat map representation of gene expression. So here every column is a patient. Patients were clustered by family history negative, family history positive, or established diabetes. And in this particular map, each row is an individual gene. So what you can see here is for this group of genes, expression was reduced in individuals with diabetes, but more importantly, in individuals with a family history of diabetes, as compared to those without a family history of diabetes. And what's interesting is that this particular group of genes were all genes that were encoded by the nucleus that encoded mitochondrial proteins within the oxidative phosphorylation or electron transport chain complexes of the mitochondrion. So all of these proteins ultimately result in generation of ATP by complex 5 of the electron transport chain. And the majority of these were decreased even in individuals at risk for diabetes.
So because mitochondrial DNA is maternal, is the maternal family history more important than the paternal family history?
That's a really good question. And the answer is that in some rare forms of diabetes, where there are mutations in mitochondrial DNA, it is maternally inherited. But that's a very small percentage of typical type 2 diabetes. Typical type 2 diabetes does not appear to have a dominant maternal inheritance, and that's probably because most of the mitochondrial proteins, the vast majority, are encoded by the nucleus. So I just showed you some gene expression changes, but if you actually measure enzymatic activity within the mitochondrion, you can see that it's decreased in diabetes. And if you look at muscle under an electron microscope, these little black oval kind of structures are all mitochondria. And you can see that in a patient with diabetes, there's much fewer mitochondria. So a variety of techniques have shown this to be a consistent defect. So mitochondria activity is also decreased in aging and obesity, as well as type 2 diabetes. And importantly, mitochondrial activity is increased with exercise and also increased with dietary and pharmacological treatments for diabetes that improve metabolism. And interestingly, mice of different strains and different resistance levels to obesity also have different levels of mitochondrial muscle activity in the mitochondria. So this may be something just to think about, that diet and physical activity may modulate whether your mitochondrial activity is low or high. So if your mitochondrial activity is low, it's associated with high body fat, increased triglycerides, and predisposition to diabetes. Whereas if your mitochondrial activity is high, either on the basis of genetics or exercise, body fat's low, and you're more likely to be relatively protected from disease. The modulators of this balance are increasingly recognized to be related to a number of genes, many more being actively discovered. But one that you may have heard about are the sirtuins. These are a family of genes that regulate acetylation of histones and are involved in control of metabolism and aging, and work in part by stimulating mitochondria, and can be activated by resveratrol, which has received a lot of attention as a natural product found in red grapes, and so thought to be perhaps linked to some of the beneficial effects of red wine. But the caveat is that if you're trying to get resveratrol from wine, you have to drink like 50 glasses a day, and so but there are actually pharmaceutical companies. There are pharmaceutical companies actually in Cambridge, one of them, that are actively looking for ligands to stimulate activity of these compounds, obviously without the alcohol. So.
I'm sorry, could you go back to that slide for a second?
Mm-hmm.
Above on the right-hand side, you have calorie restriction, and then on the left, you have metabolic syndrome.
Well, I skipped over that, but if you eat less, your mitochondrial activity improves. And there was, there's been a lot of evidence over the years that prolonged caloric restriction improves survival, improves longevity, improves insulin sensitivity. There was an article on the front page of the Boston Globe a few days ago showing a calorie-restricted monkey and a ad-lib-fed monkey, and that the calorie restriction does prolong life. And part of the mechanism may be due to improvements in oxidative metabolism at the level of mitochondria.
So on that, there's groups out there now that seem to be pretty actively engaging in advertising their low-calorie diets, and they're very thin people. Are any of those people involved in studies to look at? They are.
Yeah, there are active groups looking at those individuals. Absolutely.
What would be the caloric intake then for somebody who was calorie restricted, less than 1,800 a day?
Yeah, it's about half of what you would normally, and it's really a lot, there are different, obviously different protocols, but restriction of carbohydrates is a big part of it. Experimental at this point, but clearly overnutrition is to be avoided. So, okay, so based on these data, the so-called mitochondrial hypothesis was formulated, saying that if you had decreased activity of nuclear encoded mitochondrial genes, that could contribute to insulin resistance and diabetes risk by reducing oxidation within mitochondria, reducing lipid oxidation in mitochondria, thus increasing lipid accumulation in tissues. And so this is a hypothesis to be tested. What I've shown you and all of the data that are currently available from humans are an association. Diabetes risk, insulin resistance, impaired mitochondrial function. Doesn't mean it's causal, right? So association. So now the more difficult task is to test this hypothesis in humans It's easier to test in cells and animals, but clearly difficult in humans. So we're interested in identifying what upstream mechanisms might be responsible for this particular pattern. And so here is where we start looking at transcription and how gene expression is turned on and off. So if this is the DNA, which is a terrible drawing, I apologize, but if patients at risk for diabetes have impaired transcription of these nuclear encoded mitochondrial genes, what's causing that? Well, we know that there are a number of different transcription factors which bind to DNA and turn expression on or off, or mRNA production on or off. And so these are some of the names: nuclear respiratory factor, PPR gamma. It doesn't really matter what the names are, but transcription factors known to do that. But in fact, we looked at those, and the levels of these proteins don't differ in patients at risk for diabetes. But there's another layer of complexity on top of transcription factors. So a transcription factor binds to DNA, but then there are other proteins which are called coactivators, which bind to those transcription factors. So one of them is a family of proteins called the PGC1, which stands for PPR gamma coactivator, and there's a number of different members of the family, and this particular protein binds to a number of different transcription factors. But what's interesting is that this particular protein can, in experimental models, turn white fat to brown fat. Now, again, in the news, you might have heard recently that brown fat has been recently more well recognized as a potential source of burning extra calories. Brown fat is present in all of us. The most important locations are in the neck and upper chest and back. It's much more common, there's a lot more of it in infants and babies, but it's present in adults too. And so the reason why it's brown is actually because there are more mitochondria. So if you look at it, it looks brown, but it's really because of the mitochondria. So this particular gene can increase the number of mitochondria in fat, making it more brown. And in muscle, it changes from a type of muscle that doesn't burn fuel as efficiently as one that burns fuel more efficiently. So we looked at this particular gene in our patients, in muscle, in individuals with both diabetes and in individuals with a family history of diabetes. And you can see that the expression of this particular gene was decreased by almost 50% in patients with diabetes, but more importantly was also decreased in people at risk for diabetes. Now again, it doesn't say cause, it just says that in these individuals, this gene is down, okay? And a similar data for a related protein. So a number of years ago, then, this was our hypothesis that maybe this gene was either decreased in expression or function. That caused this impairment in mitochondrial genes, and that led to diabetes risk. It's actually a little bit more complicated than that. And whenever you see a picture like this, you know what it really means?
No one knows.
We don't know. Right, exactly. The more arrows that are on any kind of diagram, it means that we really don't know what's going on. But we've learned a lot since that initial simplistic diagram. And what we know is that diabetes is an interaction between genetics and environment. And clearly there are genetic factors that contribute to alter your body's ability to properly metabolize fuel, okay? And at the same time, there are environmental influences. So obesity or accumulation of lipid in places it shouldn't be, like your muscle and your liver, can actually turn these genes off. Even though may not be genetic, it's an environmental effect. And in some individuals, that effect will be greater than others. And that's where the gene-environment interactions may become important. But also, as you age, or if you're inactive, these things go down. So, all of these are risk factors for diabetes that are really influencing multiple components of this pathway. And in turn, decreases in expression or function of these types of genes can reduce mitochondrial gene expression, but also impair the ability of the mitochondrion to metabolize other nutrients like branched chain amino acids or fats. And we think that at this point that it's these that are probably more contributing to insulin resistance, but that's still unclear. There's some other pathways that are also important, which I won't talk about, other transcription factors as well as inflammation that contribute to insulin resistance. But like I said before, we still don't really know what's primary, and that's what we need good animal and cellular studies to evaluate. So what I sort of want to leave you with is a working model of what we think might be going on, just to give you a sense of it. So your genetic background, your ethnic background, intrauterine exposures, or aging can all reduce your capacity to oxidize nutrients appropriately. And that would be okay, but if you also have chronic fuel excess, for example, obesity or inactivity on top of that, then this impairment in mitochondrial function can cause increases in oxidative stress, accumulation of lipid because you're not burning it up, and accumulation of products of incomplete oxidation. If you can't oxidize lipids fully, you get partially oxidized lipids. And those things can contribute to insulin resistance. Unfortunately, it also perpetuates a vicious cycle because if you have oxidative stress, you can damage your mitochondrial DNA, damage proteins within the mitochondrion, reduce gene expression further, and just get a vicious cycle. So it's very challenging to try to interrupt this. Another way of thinking about it is that we need to have this balance of metabolism. So if this is a seesaw, how stable the balancing point might be influenced by your genetics, your developmental history, and your age. And we're balancing your oxidative capacity, or the number of mitochondria and their function, with the amount of food that needs to be oxidized, basically. So oxidative capacity is good, aerobic fitness is good, exercise is good. On this side, we have the load of oxidative demand that is seen at the level of the tissue, and that's driven in part by obesity, nutrient intake. So in a stable, healthy person, that's a balance. But if your oxidative capacity is impaired relative to your nutrient intake, then the balance is going to be altered, and you're going to have too much nutrient load and not enough capacity. So if you're healthy, and if you have the genes that allow you to do this, well, you might say, well, I'm getting too big. I'm going to decrease my nutrient intake and my body weight to try to reestablish a balance. Well, maybe that response is genetically determined too. Maybe some people recognize that something's awry or are able to modulate their nutrient intake where others can't, or can't do it as successfully. Whereas other people might be able to enhance their mitochondrial capacity with exercise and weight loss and establish balance by changing the left-hand side. What is clear is that if you can't compensate, then that's associated with insulin resistance and diabetes risk. So we don't understand what are the genetic determinants of your compensation or your compensatory response capacity, but I hope this gives you a sense that there's always an interaction between genetics, your underlying development, and how you respond to the environment. And we think that diabetes risk is at the center of that problem. And that's what we're trying to address. Any questions about this? Okay. So can these patterns be reversed? My patients don't seem to like this approach, so that doesn't really work very well. Weight management, lots of exercise is great. I think that one of the things that we all need to be giving the message is that exercise is absolutely critical. Not for weight loss, that's sometimes an accompaniment of exercise, but to improve physical fitness. Because physical fitness is really what's related to your oxidative capacity. And so improving fitness is where I think we've gone wrong in terms of a lot of kids that are sedentary and not getting physical education at school or at home, at home, et cetera. It's really a problem. We can use other strategies to improve insulin sensitivity, like drugs like metformin. Other therapies are in development, but not here yet. Of course, the other part of the equation is healthy diets and exercise and weight loss. Always more difficult to achieve weight loss than prevent weight gain. So what we're doing at this point is going a little bit deeper in our human samples. We're looking at metabolic intermediates, Krebs cycle intermediates, lipid oxidation intermediates, to try to determine which of these things has really altered the most in people at risk for diabetes. And you may say, well, we're not going to be doing muscle biopsies in patients. Of course not. These are research studies. But what we ultimately want to do is identify markers of insulin resistance and diabetes risk in circulating blood, which could allow a physician in an office to better identify high-risk individuals, to really hammer on, okay, lifestyle, exercise programs. We're not going to be able to fund exercise programs for everyone, but maybe we could target those really high-risk patients. And, typo, I see. How can we better target, by drug therapy, some of these pathways for better treatment of diabetes? So, take-home messages. Obesity, diabetes, and the metabolic syndrome, which is another term describing insulin resistance, are major challenges to human health worldwide. These are all very interrelated by metabolic and inflammatory pathways. And we feel that defining these links at a molecular level is the key to defining new approaches to identifying high-risk individuals for prevention and treatment. And targeting metabolic function at present by nutrition and exercise is really the way to go, and that's what we should be really emphasizing to everyone in the community, whether it's children, parents, people in the workforce. That's really the key. Not easy to do, but clearly effective. And large studies have shown that these approaches work to reducing diabetes risk. And so, it's a challenge to try to actually implement that, given limited funds and limited acceptance. But I think it has to start early, and the message needs to get out even to kids. It's important to exercise, even aside from weight management. Just physical fitness is really important. So I'll leave it at that. And any other questions?
I have a question. I'm curious, since you found all these different genes as far as what was being most effective in your gene expression profiles, are they all kind of contained to one or two chromosomes, or they're just scattered all over the place? I haven't really ever thought about this sort of thing, but I'm just curious if there's ever a global effect as far as chromatin accessibility, because of maybe these environmental factors trigger the ability to get into the chromatin in these areas, or has that been looked at all?
Yeah, no, it's a really good question. So let me first answer a question you didn't quite address, but one that I hadn't covered, which is that there have been a lot of whole genome association studies looking for polymorphisms also linked to diabetes, and these have been completed in the last couple of years. And Interestingly, there have been only a few very potent genes, essentially one, which may influence the body's responsiveness to insulin, secretion of insulin in response to food intake. But there's a problem with some of these genetic studies in that they're looking at patients with diabetes. So by definition, those individuals have already failed their insulin secretion. But what's really surprising is that there's very few major findings that are linked to diabetes susceptibility. Again, highlighting the fact that it's the environmental influence on the background that's key. And so obviously those studies are ongoing. Now, in terms of your question about the genes that are affected, for example, the top-ranking genes that are affected are all scattered around different chromosomes. They all tend to be coordinately regulated by a couple of dominant transcription factors. But again, that suggests that it's an environmental influence on this group of genes rather than a single genetic locus that's responsible. One sort of regulatory mechanism for gene expression is modification of histones and unwinding of chromatin to make it accessible to transcription factors. And I think that that's clearly recognized as an important regulatory step. Some of the genes that have been implicated in mediating some of these patterns are clearly responsive to drugs that change chromatin structure. And the sirtuins, the resveratrol, grape, red wine link, is actually, a set of genes that actually can affect histone modification. So I think that that's a really interesting candidate group of genes for mediating some of these effects. They can be regulated by nutrients. So, really good question.
So, as teachers, what is the next step? I mean, how does this go from research into public policy? It seems to me, at least at the schools I've been to, they feed the kids to get diabetes. I mean, there's so little nutrient value and it's so high in sugar and fat. How do you, I don't know if you have any suggestions, like, what can we do with this information to get back to our schools and...
Well, I think, I have 3 kids, and I'm appalled by the stuff that comes to soccer games and comes to baseball games. The kids run around for 5 minutes and you have to have a snack, which is a donut. I mean, it's ludicrous. And it's really hard to walk a tightrope of, not being too negative all the time about certain food choices and to be positive. But I think we need to sort of just be advocates vocally for improving nutrition, making sure that there are always healthy choices available. And the reality is that that does work at some level. I mean, soda machines are slowly getting out of schools, and that was the work of a few devoted people, and I think we just need to keep hammering at it. It's going to take a while to get the message across. We can't be draconian and say, we'll never eat another French fry in our life, but, sort of trying to spread the message that this is not a personal health issue, it's a public health issue. And just like we all work to stop the spread of tuberculosis, we have to work together to stop the spread of this environment that doesn't help us manage our metabolism.
Well, I guess they got rid of trans fats. They can do this.
No, that's good. And I think we have to chip away at it and be positive, but maybe the positive message is easier to promote. Healthy living, exercise, lots of vegetables, trying to, the other thing that I sometimes, I try different approaches with patients, try not to eat something that a bunny wouldn't eat. What do bunnies eat? They go in a garden and they eat all the vegetables you're trying to grow. That's the stuff that is always healthy. That does not do anything bad to anything. Don't eat anything your grandmother wouldn't eat. does your grandmother know what a box of, or maybe your great grandmother, you know, a box of processed crackers with saturated fat filling in between the crackers. You know, processed boxed food is not a high fiber, whole grain choice. And I'm not saying that all boxed foods are bad, don't get me wrong. But it's complex carbohydrates are missing from our diet, and a lot of simple carbohydrates that are quickly absorbed, stimulate high levels of insulin production, facilitate deposition of fat is not a good thing. And so good choices are probably things that are not as processed. Not that I have any objection to the processing itself, but the net effect is not a complex carbohydrate. And so, you know, we have to start slowly moving toward what we used to eat, which is when we were a lot healthier. You know, it's not easy. I don't have any. I think something like the Surgeon General can really, a role like that could really do a lot for trying to sort of walk the fine line between messages and public policy. That's kind of the level where I think it ultimately will, but individual, local level is really important.
I was wondering about that, one of those first slides you showed about the map of, so if it is a global epidemic, do the same things apply? Like, is obesity increasing by leaps and bounds in China and Africa and Mexico?
Yeah, so what's really interesting is that there's different thresholds. So for example, in Asia, the threshold for obesity is much lower. So a good example is South Asia, India, the Indian subcontinent. So there, a little bit of abdominal obesity is much worse than for a Caucasian. So a body mass index of 25 to 27 would be considered, you know, okay here, not great, but okay. But there, it's really deleterious. So the ethnic background may influence your responsiveness to how much fat you deposit in an overnutrition setting. So it seems that in Asia in particular, the thresholds for adverse effects of obesity are much lower.